Thank you everyone for joining this presentation. So what is diversity? Diversity is a term that normally is used for human, and I will speak about preclinical studies. So diversity as a definition in any dimension that can be used to differentiate groups and people from one another. And this can be related to age, disabilities, weight, sex, ethnics, religion, sexual orientation, education, and national origin. This is a big issue now in clinical trials, and how and what way can we address in preclinical. So we most definitely can refer to age, to disabilities and comorbidity, weight, obviously, and sex. And I'm going to give an examples on all three of those that can maybe shed a light on how this might be addressed in the preclinical studies. We do believe that diversity should be taken into account and under consideration already when we are talking on the in vitro studies. And I will use an example in vitro that is age sensitive, and all the way to large animals. In rodents, I will speak more about stroke and comorbidity. And in the pigs, I will refer on sex differences in pain. So let's just start. The first example is an in vitro age-sensitive assays. You know, when you talk about spinal cord injury, for example, so what you actually do, you take a piece of the spinal cord, which is actually the injury because you take out the spine and then you activate all the components that are leading to the skull formation. And then you plate the tissue and you look at neurite outgrowth, synaptic formation and such. And by pulling or taking out the piece of the spine, you actually triggers a lot of components that are inhibiting the neurite outgrowth, which are the GAGs and the NOGO-A. And they are responsible for the fact that the nerves and the neurites will not grow, and later on the skull formation is created, and that blocks the ability of the nerves to grow and cross the skull formation. In most cases, when it comes to study spinal cord injury, they would use animal models that are referring to newborns, and then they would take the spine, plate it, and then one day after the plating, this is the culture that you get. There are a lot of neurites, a lot of connections, and a lot of communication. And the reason is that the GAGs and the NOGO-A are non-existing in the newborn rats. So actually we are missing the point of looking at neurite outgrowth at a situation of spinal cord injury in adults. But this model is most commonly run and used because of the easy way of doing it, and because it is very much available and easy to do. However, if you look at a spinal cord that was harvested from an adult animal, what you would see is neurons here that do not grow or have very little neurites. And this is actually the picture that you're going to get also in the in vivo and in human. So it takes a little bit longer, but this is the situation that one might to look at. And here it is, when untreated animals, as I said, after 10 days, you would see the same pictures. This is the neurites, neurons, and oligodendrocytes. If you expose it to a positive control, in these days it's copoxone, then you would see much more developed neurites, longer neurites, which you can measure. And if you stain it with beta-tubulin 3, then you can also detect the synapses along the neurites. And then if you compare the positive control with the very long neurites to the vehicle, you would see that there is no neurite outgrowth. So this is reflecting the real situation of adult injury in spinal cord, although it's a longer study, but I think it's more relevant. And this is only one example for age-sensitive assay. And then you can do all the calculation and quantify this assay. And what you would find is that with neurite length, per cell, there is an active positive control, which is the copoxone. It's increased the neuric index, and it also increased the synapses per cell. So you can learn quite a lot from this assay, not just about the neuric length, but also synapses. You can also look at synapses and synapse strengths and learn if it is a excitatory or inhibitory synapses, of course, by using different staining. So this is already taking into account age-related assay as a part of diversity. If you go to in vivo, situation gets, of course, more complicated. And with the stroke here, this is one of the models that I chose as an example, but of course, this is correct for any other assays that we are doing. And so what is between the strokes, rodent studies and clinical studies? There are three components here. One is the methodology, and of course, including diversity. Another one is comorbidity, which is part of diversity, and the endpoint and outcome. I will speak about all those three components, but I will be more focusing on the comorbidity. So stroke, we think about stroke as a model where there is a block of the blood delivery to the tissue, to the brain. And this is how a clinical study would look like. We will look at older population because most stroke events are happening in older population. We'll look at diverse population, different races, and also different sexes, men and women. And most of them will have some kind of comorbidity, whether it is obesity, diabetes, hypertension, or other comorbidities. And obviously, they will all take drugs to reduce those and control those comorbidities. But when we will go to the preclinical studies, this is how it will look like. And what we will see is actually the studies will be in young, naive animals, usually male, all from the same strain, all from the same weight, or as possible, as from the same weight as possible. And you can see the difference in the choosing the animals and how it is in the clinics. And then we want to translate something that is very, very unique to the way we look at preclinical and simplifying the situation. And then from a simple solution, we want to predict what is happening in a more complex disease. And as you see here, the system in the rats, as I said, would be very young and old, sex normally male, hardly any female. I can say that if you look for studies that are done in stroke, there are 3 times or 4 times more studies in male than female. And medical history or none, because they're all healthy animals, and the outcome almost predominantly would be infract size, whereas in clinical trials, we obviously have different demographic and clinical characterization. Age is adult, sex it's female and male, and it's mixed. The race will be any race. Medical history, all or most of them will have comorbidities, and the actual outcome would be functionalities. Now, had we not known that this is all about stroke, and I would have shown you the left part on the rodent here and the right part on the human, one might think that we are talking about 2 different diseases. And indeed, in a very, very nice review from last year, the authors summarizing why therapeutic of acute stroke study were positive in rodents, but completely negative in patients. On A here on the left, they show the percentage of studies that are reporting infract volume. And you can see that in experimental studies in rodents, it's 100% infract size, whereas in the early clinical studies and late clinical studies, it's very low, it's less than 20%. And the studies that are reporting functionality is about 50 to 60% in the experimental studies, but almost 100% in the clinical trials. So the clinical trials are actually looking at functional outcome, and experimental studies are actually looking at the infarct studies, infarct outcomes. So they both look at different outcomes. The same goes on the methodology. The 100% of the early clinical and clinical phase are reporting randomization, whereas in the preclinical studies, only 50% are reporting randomization. For blindness or blinded outcome assessment, then on the phase 3 clinical trials, it's almost 100%. In the experimental group, it's not, it's 50%. And for functionality, is exactly the opposite. Most of the clinical trials are involving one kind or two kinds of comorbidities, where the experimental studies are all done in naive animals. So if we conclude that, so in opposed to clinical trials, preclinical studies are normally focusing on infarct size, no comorbidity, and less randomization and blinding. Now, does it matter? Maybe it doesn't matter. So let's see a drug that was tested and was found positive in normal stroke study, but not so positive when comorbidity was involved. The model that was used was a 4-vessel occlusion model. It's a model of which the vertebral arteries are permanently occluded, and carotid arteries are occluded for 15 minutes, which creates a specific hippocampus injury. You can see in the histology pictures here. On the upper A panel, what you can see is almost non-CA1 cells, pyramidal cells, whereas in naive animals, this is how it looks with a lot of pyramidal cells. So this is a very typical outcome of the 4-vessel occlusion model. So obviously, one of the things that one would look at when the hippocampus is injured is the water maze assay, where the animals are introduced to a pool with a hidden platform, and the animal is learning to find this hidden platform. And the first phase of the hidden platform, the animal should swim directly to the platform. Let's see how it looks when the animal is not learning. So this is the red point is the animals, and for 1 minute it just keeps swimming. And even though the animal already was introduced 8 times to the water maze, the animal does not learn and does not remember where the hidden platform is. And we can just go on and swim, and you can see here that the animal was very close to the hidden platform, but it did not climb the platform. And then the animals keep swimming, passing just by the hidden platform and not climbing it. And following a drug treatment, the animal did learn how to get to the hidden platform and immediately swam to the hidden platform and climbed on it. If we do the quantification and the statistics, and this is how it looks like. The sham-operated animals learn, this is the red line, and on the x-axis is the number of trials, and this is the time in seconds for the animals to find the hidden platform. You would see that gradually this time decreases until the animals swim directly to the hidden platform. With placebo and 4-vessel occlusion, you see that the animal did not find the hidden platform. And with the test item, you may see that it was as close as it's possible to the sham, and the animal gradually found the hidden platform. This was all very nice. It was a stroke in healthy adult animals. Everyone got really excited. And then we added to this the diabetes. And we ran the same study, same experiments, same test, but the animals were diabetic animals. And these were the results. In this case, the sham animals did swim nicely and found the hidden platform, but the test item treated animals were as good as the placebo, and they did not find the hidden platform, meaning that under comorbidity, the results were very, very different from the study that was run in adult healthy animals. Now, I need to say that diabetes is only one comorbidity. There are also hypertension and aged animal. There is studies that were run in aged animals and in aged rats, and this is quite interesting because in aged rat, actually there was 2 or 3 publications that shows improvement because of the collaterals that were built with age, and also hypertension. There are studies that were run under the background of hypertension, and I think it is very, very important that studies will run with comorbidity. The last example that I want to present here, and then I'll be very happy to take questions, is sex differences. And that's the last diversity that I want to speak about, and it's related to pain. And you know, the myth is that women can bear pain better than men because we are giving birth, and that's painful. But this is actually just a myth. In reality, women are less tolerant to pain than men. And actually, they are simply different. Maybe when it comes to chronic pains, we know that in chronic pain, the pain is mediated by microglia in the spinal cord, whereas in the female or in women, the pain is mediated by T cells in the spinal cord. Now, if you do a search in PubMed and you cross pain and microglia, in the last 5 years, you get more than 1,000 papers. If you do the same search on pain and T cells in the last 5 years, when it's known that T cells are participating in the pain process of chronic pain, you would find less than 400. So the sex difference matters, but it's not addressed. It's addressed in the clinical study, but not in the preclinical studies. And if you look here at the percentage of male and female in postoperative pain in rodents, you will see that really the vast majority of the studies were in male here, and a lot less in female. And it gets to the point that McGill, who is one of the leaders in this area, suggests that the entire existing preclinical pain literature may be male biased. And if you look, but there are also some positive in this presentation, especially if you look at this part of the graph. On the left part, it's the number of sex differences studies over the years. It's increasing dramatically in 2012. And in B, it's the chronic and reporting of animal subject in Journal of Pain. And you would see that the blue here is male only. So still at the beginning of the 2000s, it was male only, but with years, the blue part of the column is getting smaller. Female only is also, it's there, but for me, it's the same. If it's male only and female only, it's the same. Male and female, but no analysis on the sex difference is very thin here. And male and female, no differences. It's important to know with which models we don't have difference and when we do report difference. So the number of studies that are looking at female and males and are looking at the differences and taking and closer look on the difference between the female and male is increasing. And by that, to my opinions, decreasing the chances for failure in the clinical study. I want to share with you some data that we have on post-operative pain models in pigs. This is the skin and muscle retraction model in pigs, where we do an incision on the area of the flank. And this is where we measure the pain using von Frey filaments. For those of you who are not familiar with the von Frey, it's filaments that produces pressure on the area of which is applied, and the level of pressure increases, and you can change the filaments. to increase the pressure. So the higher the pressure you need to apply for get a reaction, the less pain it is. So if we look at the left graph here, so this is the area of, I would say, pain, because you see that we need to apply really 0.16 grams to get a reaction, and this is the area of no pain when one needs to apply 60 grams to get a reaction. On the y-axis is the percentage of animals that responded. And this is even before we did any surgery, and one can see on the red line, which is the female, that females already responding to smaller filaments than the male. The female is between, let's say, 8 to some of them 60. In the male, it's 15 to 60. 2 hours after the incision, here it's really different, and we would see that most of the animals are actually here in between 0.16 grams to 0.6 grams. And in the male, it's around 0.6 gram to 6 gram here in blue. So there are differences between the female and male here, where female were more sensitive. And we asked ourselves if that has anything to do with the fiber density, skin fiber density. And it happens to be that the answer is yes. And if you look at the intraepidermal nerve density of the female, it's higher significantly than the male. And this is also what we find in women. And yet, most of the studies are run in male. So what about locomotor assays in response to different drugs? So we can say that female and male pigs respond differently to drugs. And in this case, we looked at the behavior of the female and male to morphine. And on the right, the male, this was published. What we see is that the pre-dosing, the animal would walk in the open field about 60 meters, and that increases with morphine. And in higher doses, the animals even becomes very aggressive. But in the female, we don't see the same phenomena. We see more or less the same. They walk more, but we see the same walking distance as a response to 1 and 5 milligrams per kilogram. There are no different statistical differences here. So they are also responding to drugs in a different way. They are less sensitive to morphine than the pigs, and that is also the case in the human. So to conclude, before I get questions, as we believe that diversity consideration apply early when designing the in vitro studies is very important. Addressing sex, age, comorbidity, weight, while planning the efficacy studies can save time and money when planning the clinical studies. And by that, I would like to thank you all.